Um, all right, so hi everyone. My name is Zahra, and myself and Ahmad are bioinformaticians at the European Nucleotide Archive, so based at the EBI. And today we're going to be talking about automating a COVID sequence submission tool, and how and why we decided to automate this using Nextflow. But first, who are the ENA? So for anyone who's unfamiliar, the ENA is an open access global nucleotide sequence repository. We cover raw sequence data, sequence assembly information, and functional annotation for anything that is non-human. We're the European arm of this international nucleotide sequence database collaboration, which means we mirror the data hosted by our sister nodes. So one of which is called DDBJ, based in Japan, and the other is NCBI, based in the US. So the link between SARS-CoV-2 data and ENA. Well, we received over the pandemic an unprecedented volume of SARS-CoV-2 data submissions, and this really picked up from March 2020, such that now SARS-CoV-2 data submissions to the ENA make up 24% of all ENA raw read data. And this is volumes in the millions of records. And you can see this from the graph on the left, or the two graphs on the left. So we have one for nucleotide sequences and then one for raw reads. And you can see we've got over 6 million of each. Now, all of this SARS-CoV-2 data that's submitted to our archive, when it goes public, it then presents in our ENA browser, and that is then fed, and all that SARS-CoV-2 data is fed into a dedicated COVID-19 data portal. And this is really a focal point for not only sharing public sequence data for COVID, but a variety of other COVID-19 related data types to the scientific community. So I wanted to include this slide to show the spread of COVID data submissions to us. You can see it's very much global, but we do tend to have a high density of submissions from Europe. We are, though, actively engaging with low and middle income countries now. You can see that the picture is very much similar for raw reads and assembled sequence submissions. And our submitters tend to be, well, can be either those that are part of a national sequencing effort. Also, they might be individual submitters. So these are people from research labs or hospitals across different regions. So I alluded to the COVID-19 data portal being a central point of access for all types of SARS-CoV-2 related data for the scientific community, but I wanted to highlight how it's one component of a wider ecosystem of tools and workflows and engagement on a national level, which makes up the European COVID-19 data platform. And this data platform was launched in 2020, and the main aim of it is to facilitate rapid data sharing, storage, and access for SARS-CoV-2 data. And one entry point into this entire data platform is through SARS-CoV-2 viral submission tools. But before I talk about the different submission tools that we have, I wanted to quickly go through the ENA metadata model, which describes how ENA submissions are structured. So firstly, we have a study object. This will hold information about, well, all of these boxes below. So information about the sample. So this will refer to your COVID-19 virus, so the sequenced biomaterial. So for example, how and where it was sequenced, how and where the sample was collected rather. We have then the experiment, which contains metadata about the sequencing method. The run object, which contains which holds the data files for your raw reads, and then finally an analysis object, which contains metadata and data files about the assembly sequencing method, or the assembly files themselves. And I've just included numbers here to indicate that each of these objects are submitted in a sequential order. So this is what any ENA submission and all SARS-CoV-2 submissions to us would look like. Now, how can users submit SARS-CoV-2 data to us currently? We have a number of different methods that are suited to different volumes of data as part of the submission, and also different skill sets. So we have a browser-based method that uses spreadsheets. We have a web-in command line program that users can download and run from the terminal. We also have another programmatic route that involves the creation of XML or JSON files that can then be submitted via curl. However, each of these has their own limitations. So the browser-based method is for metadata submission only, not the actual data file itself. WebInCli and the XML and JSON-based method, they require a certain level of technical proficiency, which often with submitters who are, say, wet lab scientists is not always the case. And if you have submitters who would like to publish a paper, and that's the reason why they're submitting data, this can often be a slight barrier to data submission, which is why we developed, or the ENA and the AIT team developed this SARS-CoV-2 drag and drop uploader tool, which offers an alternative route to submit data that's got an easy to use and simple interface, which involves a user just simply dragging and dropping data files and a metadata spreadsheet with a secure key. And the secure key is given by bioinformaticians like myself and Ahmad to the user. You can see the file extensions that are supported. I will just show a demo now of the front end as to how this works. Okay, so you can see you just enter your secure key that's provided to you as a user. You enter your name, your email address, and also this web and submissions account, which is your ENA submission account. It's if you don't have one, there's a link to from the platform as to how to register it. The next step is to simply drag and drop your data file. So in this case, we're using raw reads, FASTQ files, and the accompanying metadata spreadsheet. You click the consent box and you press upload. Once you've done that, you'll see a list of all the files that have been uploaded. You can add a note as well regarding your submission, which I seem to have typed quite slowly. But once you've written that, you just press submit, and the bioinformaticians at the ENA are then notified by email that a submission has been made. And you can see here, this is a list of what the user has submitted to their particular upload area. But now I'm going to pass over to my colleague Ahmed to discuss the backend.
Thank you, Zahra. So my name is Ahmad, and I'm gonna walk you through the backend development and the automation of this tool. So in the beginning, we used to, the backend of the tool was basically manual. So it was a collection of scripts. These scripts were like, we just run it by ourselves or just manually in the backend. So after the user upload their data to the S3 bucket, so we retrieve the metadata, we inspect it manually. Then we verify that the data is correct or not. Then we start parsing the data, creating XMLs and submitting the samples and the studies. Then after that, we move on to the genetic data, which is the data files. We submit them using WebInCli. Then we confirm that everything's went through, and then we notify the submitters. It's a bit of a long process. So we try to add a wrapper, which is a Nextflow, to automate the whole this collection of. So what we've done basically that after the user submit the data or upload the data to the S3 bucket, we trigger the Nextflow pipeline. So the files will be transferred into the EBI cluster. We call it code on. And then the file's integrity will start to be validated. Then after this validation, it will move through, the metadata will be fetched and will be parsed. The spreadsheet will be parsed to studies and samples. Then we create XMLs and it will be submitted into ENA as sample and studies. Then the script will wait to be informed from the ENA if the submit went through or not. In case of an error, it will go to an emailer script, trigger an emailer script that will send an email to correct this error. Otherwise, if it is successful, it will go through to the next step, which is submitting the data files. So it will take the data files and submit them through the WebInCli software. Of course, in case of success, we'll inform as well in an email or Otherwise, there's an error. Also, that same thing will cause a trigger to the emailer script and will send an email to trigger the resubmission again to fix, of course, the error and resubmit again. So why did we actually use Nextflow in this pipeline? So Nextflow, it's a really good program, firstly, and has a lot of features, but we mainly used it for these specific points. So first of all, it's the modularization feature. So it was really beneficial for us that you have a module for each workflow. This module, you can use it as a separate workflow, or you can transfer it from workflow to another workflow. Also, of course, the separate work directory, which was make your life really easier for debugging. For each process, it has its own work directory. But actually, the hallmark why we did use the Nextflow is actually the support. There is a massive collaborative community in this for Nextflow. It was really helpful. There's a lot of events, there's a lot of tutorials, a lot of trainings. Also, there's a lot of examples pipeline that you can look into, you can take from, and stuff like that. And also, of course, we can't forget the documentation. There is a really good documentation for Nextflow. Finally, which is a bit not exclusive for Nextflow, you can find it in other workflows, but we found it really easier in Nextflow itself and more easy to comprehend, which is channel output indexing. Which means that there is multiple output for one channel, but you can use one of this output to another channel or another process, and the other output for another process. Of course, it's found in other workflow, but for us, we found it easier for us to understand and easier to comprehend in Nextflow. The pipeline. So actually, we created two pipelines, not one pipeline. One pipe is the back-end pipeline, which is what we are actually talking about now, linked to the front end in the drag and drop tool. But we created another pipeline, which is standalone pipeline, which is a pipeline that the user can take and run in their own local environment. Without the need of it doesn't have a transfer step. So both pipeline mainly identical or similar, except there is no transfer step. So we don't transfer the data from S3 Pocket to Codon. So they just run it in their own local environment. They just only have to add the path for the spreadsheet and the data file. But the rest is mainly identical. So after the upload, the S3 Pocket look like that. So it has, it's not clear here, basically, but it has here the user ID that we provided them. It will be created as a directory. Inside this directory, you will have the file that the user uploaded. So that's how it looks like. So after that, I'm going to show you in details how it looks like inside in a bit. But after that, there is two routes. After we trigger the Nextflow, there is two routes. We have either partial submission or complete submission. Partial submission, which is in case there is an error happen in the metadata. So the metadata is not good. It's not validated correctly. So there is a problem with the metadata itself, sample or study. So here in this case, you can see here, it will transfer the data. So that's the transfer step. The second step, which is the metadata submission, which is the validation of the metadata. So if there is an error here, it will hold the pipeline, it will stop there and skip to the metadata email, send an email to fix that error and resubmit again. Otherwise, if a full submission, there's nothing wrong with it. It will continue to what they call it bulk web initialize submission, which is a software, which is the process to submit the data file itself. Then we'll move to an emailer script to notify by email. I'm just going to show us a really brief demo how actually we see stuff there. So you can see here, this is the S3 Pocket actually that we actually uploaded the data now. Zahra uploaded the data in. So that's this one of the directory. That's the actual directory, the user ID, and you can see it has these files inside it and the metadata file. So after that, we actually go to just to show it really clearer. So here we go to we trigger the Nextflow here in the Codon or API cluster. So after we trigger that, you can see here, it's going to take a bit of time, but you can see here, Nextflow has multiple parameters. You have the Webin account parameter you need to add, which is the Webin account, of course, it is the account for the user in ENA. They can create it. Every user has its unique ID or your account. We call it weapon. So they need to provide it. The context, it's read or genome. Mode, which is submit or validate. This is UUID, which is the user ID, that's the user ID. And of course, the environment, which is test or prod or so. So you can see here the first script, which is run through, which is the transfer integrity. So they transfer the data from S3 bucket to here. Here's the metadata submission. So it went through. So they validated the data is correct. So it went through, and see, I'm receiving emails now. So then you have a trigger them emailer script, which is this the third step. Then the fourth step, which is this one, which is validating the data, submitting the data files. Then the final one, which is receiving the receipt from the weapon client, if everything correct or there is something wrong. So how the receipts look like. So these are examples of the receipts. You can see here the first one. I'm not sure if they're clear enough, but we can see in the first one the sample error. So here is an error in the sample submission. You can see here we send them what is the value, what's the problem with the metadata, where's the problem, and we suggest a solution for it as well. And here you see a weapon client receipt, which is the problem. If there is an error in the weapon client, and the error in the data file, also it tell you also what type of, what's wrong with the file, which file has a problem. And you can see here the successful submission. You can see from successful submission here, you have success is equal to true, and it will give you the accessions that actually been submitted for each sample or project of run. So the limitation of our project, of our tool now. So the tool is currently under development. We're still working on it. We're still developing. We're still debugging. So until now, the tool does not trigger automatically. So when the user uploads the data to the S3 bucket, it doesn't trigger. You need to trigger it manually. You need to press the Nextflow trigger that as I've done to run the software. The second one, the partial submission. Partial submission, it still have problems. So if there's half of the samples been submitted, the other didn't submit, you need to make some tweaks in the metadata to overcome this. So it's still not as flexible as we wanted. We are working on that as well. And also submission reads and associated genome or gene associated read together in one run is still limited to that. We don't do that yet. So you need to run it twice. You need to submit the read first, then you submit the genomes after that in two separate runs, not in one run. And finally, this tool is SARS-CoV-2 specific. It's COVID specific. We are trying to expand it, but until now, it is still SARS-CoV-2 specific. The future plans. So firstly, we are presenting the same tool in the summit, the Nextflow summit, that's going to happen in October this year. We have the feature improvement as well. We have short term. The short term, firstly, as I mentioned earlier, we are trying to merge the submission of the genomes and their associated read in one metadata in one run immediately without the need to run it twice and change the metadata in any way. The second one is to tackle the problem of partial submission. So still we have some issues with that, and we are trying to find a solution to work around it. And of course, one of the important part, which is adding a Docker or Singularity environment on our script, especially to the standalone script, because this script is going to be transferred to the user local machine. So it needs to transfer as well the environment with it. So it still hasn't been done yet, but this is a short-term goal, and we hope to do it as soon as possible. And we have some also long-term goals. We are trying to improve the standards in the script just to try to share it and put it in one of the repositories such as NFCore or Workflow Hub. And finally, to expand the species for this tool to more than SARS-CoV-2, at least to cover the viral species. And thank you very much for listening. I would like to thank everyone who has really contributed to this work, especially the people that's mentioned in this slide. And thank you everyone for listening. And I'm happy, and Zahra, of course, to answer any of your questions.